So it's my absolute pleasure to introduce Dr. Robert Gerszten as our first speaker of the year for Cardiology Grand Rounds. As many know, Dr. Gerszten is the chief of our Division of Cardiovascular Medicine at BI, as well as the Herman Dana Professor of Medicine at Harvard Medical School. I also have the pleasure of knowing Rob as a close mentor and friend for 14 years, which is a number that probably makes us both feel old at this point. So Dr. Gerszten did his clinical training at Johns Hopkins, Penn, Mass General Hospital, as well as intensive research training at UCSF and Mass General. And he's developed a large and very innovative program using translational omics techniques to understand mechanisms of cardiometabolic disease, which we'll be hearing about today. Dr. Gerszten has received a number of awards for his research, including an Established Investigator Award from the AHA, and more recently, a Lifetime Mentorship Award from Harvard Medical School. And so we are absolutely delighted to have Dr. Gerszten present today on 120 terabytes of genomics data to gain insights into cardiovascular disease. So thank you, Rob.
So good morning, everyone. I haven't ever given grand rounds without the kind of interpersonal interaction. I do have Joe, fortunately, Joe Canham's kind of socially distant away in my office. So I do have a couple jokes, and I'm hoping to at least get some reaction as to whether they're good or not. Joe will give me an honest opinion during the presentation. So the title of the talk recently came up recently during a lab meeting. And we were talking quite seriously at first, about how we were going to handle a bunch of data that we had just generated. And then Dan Katz in the group began to muse a little bit. This is, you can see the slide here. This is just, it doesn't really matter. It's just a line of data from what looks like even, could be an Excel spreadsheet or whatever the readout is here. And it was just looking at an association between one part of the genome and some protein. And then he began to muse about, how much data we had just generated and are generating as part of these big analyses that we're doing. And he said, gosh, we're looking at 3 billion base pairs. And just to make it a little bit more tractable, he started off by looking at 28 million variants in the human genome that were available at the time that you could see in at least a couple individuals, okay? And that's like 5 gigabytes of data. We were looking at another data set. I'll go into all the names later, but in which we had 18 million variants, and that was about 2 gigabytes of data. And then we were looking first at the Jackson Heart Study data, and we had measured 1,300 proteins and 3,300 metabolites. We were also looking at the multi-ethnic study of atherosclerosis, in which we measured about 1,300 proteins and 3,300 metabolites too, but we've actually done it at a couple time points. So we've looked at protein changes, or we're looking at protein changes, we're looking at metabolite changes. And it turns out this heritage study, we've measured 5,000 proteins and 5,000, but these individuals have undergone an intervention, an exercise intervention. So we're looking at 5,000 protein changes and 3,000 metabolites and 3,000 metabolite changes. And when we kind of have looked at the first set of data, gosh, it's 125 terabytes of data that we've accumulated in the group. And just to put it in context, Dan said, well, what's out there? What do we know? Well, it turns out on the left is the Library of Congress, and that's actually only 20 terabytes of data. On the right is the entire Twitter sphere, and that's 140 terabytes of data. So with what we've done even more recently, I think we've kind of surpassed that much information. Now, not every, we're kind of proud of this. Not everyone is as enthusiastic as we are. In fact, some of you might have seen a tweet this morning from the president that I had more people attend my rally in North Carolina this week. Okay. Now, I want to, highlight we're going to highlight much of our own work, but I do want to say that there are a number of groups in the division that are looking at extraordinarily large data sets and turning that data into human knowledge. This is, I'm going to show some examples from the Smith Center, for example, Eric Sezemski and Cheng Yu Shen and Bobby are working with 168,000 patients who have undergone a variety of, in this case, the SAFE PAD study, but looking at interventions, and this is work with the FDA to look at outcomes in those individuals. Rishi, Wadhira, and others in the group are looking at a large CMS data set that in this case, for this example, has 8 million index hospitalizations for acute myocardial infarction, heart failure, pneumonia, et cetera, some of the big outcomes that we look at in lots of data sets. And, and then finally, Bobby's group and Kazi and Rishi are working with Cheng Yu and the American Heart Association and become a core to analyze what will be about 15,000 COVID-19 hospitalized patients. So enormous amount of data there, a little bit different sort of data. And then similarly, Mike Gibson is leading a study with Apple. This is the Heartline study that will include almost 150,000 individuals to use devices to study atrial fibrillation. And, we were kind of bragging with 100 and some terabytes of data. They're actually going to be generating 6 petabytes of data from these individuals. Although I will humbly submit that, number one, the per person amount of data we're generated is actually higher in our analyses. And also, our lab is slightly smaller. We're a small cap company compared to a $2 trillion Apple company, but really remarkably large data sets that are going on at the rest of the division. So what am I going to talk about for the next 45 minutes or so? So, after a little more introduction, I'm going to discuss a little bit, update you on the nuts and bolts data generation for large multi-omic studies in which we're participating, particularly the NHLBI sort of premier precision medicine program. It's called the Trans-Omic Precision Medicine Program, or TOPMed. I'm also going to tell you about what is probably the per person richest data set that's going to be ever established, at least right now, called MoTrPac. I'll tell you about that. And then more importantly, we're going to have like a pivot in the middle of the talk and say, well, so what, right? I mean, show me some applications of how we can turn these measurements in the blood, for example. I'm going to pick some select parts of the omics slice of the pie and maybe integrate that with genetics. And then conversely, think about how this information could be used to illuminate what's going on in hotspots in the genome that we or other people have described to add meaning to those genetic areas. And what I ask is that you step back and think of blood LDL in your mind. What would it be interesting, and this has of course already been done, but would it be interesting to understand the genetics of blood LDL? Well, it certainly was for Brown and Goldstein, and for it's turned out that way. Now can we do this for 7,000 elements that are, or 10,000 elements really, that are circulating in the blood? And then I'm going to give you a little bit of thought about how we're really, I'm going to pick out a few examples, but then step back and say, how are we prioritizing this in a formal way? How are we triaging? And what are our future directions? Okay, so just by way of reminder, DNA is just the blueprint, okay? In 1953, I think it was, or 54, when Watson and Crick established the biochemical characteristics of DNA, we thought, is that everything, right? But we know that that's just the blueprint for a house, and we really need to understand the bricks, the wood, and how it's all put together. And many of you have seen these slides, but I want to catch everyone up and put them at the same position because there's lots of new data here as well. But just by way of a reminder, the central dogma, is starting to evolve. So this is over 10 years ago when Tommy Wang and I were asked to write a piece for Nature where we might mine for new disease biomarkers and biological pathways. We just highlighted that genes make transcripts, transcripts make proteins, those proteins that are enzymes make metabolites. And we posited that, this information over here on this side of the slide is particularly close to any given phenotype. And because, as many of you seen this slide before, it integrates environmental cues, diet, exercise, et cetera, it might be particularly important for disease biomarkers. And now we're integrating all that information. So that was maybe 10 years ago. And in fact, the central dogma of biology continues to change, right? I showed you all those things moving forward, you know, from genes, and now there's a lot of work in the epigenome as well, and I've already mentioned the transcriptome and proteome, but what we're learning is there's a lot of feedback loops as well. So for example, there are metabolites in cancer that actually, these oncometabolites, these butyrate species that actually modulate transcription. And there's even thoughts in cardiology. Heinrich Tagmeyer at the University of Texas, a wonderful, you know, father of cardiac metabolism, has shown that there are metabolites that actually modulate cardiac function in this sort of in transcription, this sort of feedback loop as well. So it's eminently complicated. But I think it really behooves us to get as much information as we can and integrate it as logically as we can as well. Okay. Now, a lot of people are highlighting the role of genetics, okay, including several of our colleagues and our collaborators, several of whom I had a call with yesterday. And there's no question that genetics is the cornerstone of all of this. But in a recently published paper over the course of the last year, Tommy and I, Thomas Wang, who's now the chair of medicine at UT Southwestern, Jonathan Mosley, who's a fantastic investigator at Vanderbilt, Deepak Gupta, and others, you know, we began to look at this information from the polygenic risk scores and then thought about how about all these other types of omics information. That's the work that we're doing. What adds more to disease prediction, and how about just clinical values as well. I'm not going to go through all the details. You can kind of stare at the line of C statistics that don't vary that much when you add genetics on top of basic kind of models that include these clinical factors. And in fact, if you do other types of investigate other types of metrics of disease prediction, such as reclassification, it turns out that only 5% of individuals reclassified using a polygenic risk score, and for those having events, 80% of the reclassifications were actually incorrect. So I am certainly not saying that polygenic risk scores aren't going to be extremely useful for certain cases. Absolutely. What I'm trying to tell you today is that that other information is also going to be equally important. So what is this program in this Trans-Omic program for precision medicine, or this TOPMed program at the NHLBI? Well, I took several of these slides, or adopted them from a recent presentation that I participated in with Wayne Gann, who's the PI for the NHLBI program officer for this program. And I'm going to highlight our participation in here. And in particular, I want to highlight the work of Debbie No, who's a pulmonologist, Lori Farrell, who's been working with me for between 15 and 20 years now, Zhu Shi in our group, and Xuning Zhang, who developed a lot of the data that I'm going to show you today as well. So this is the largest whole genome sequencing data set in the world, okay? And it's the largest multi-omics data set in the world as well. So what is it comprised of? Well, it consists now of about 155,000 participants from about 80 different studies of varying designs, okay? And this slide highlights that it really is an equal opportunity event for anyone interested in cardiac, metabolic, pulmonary, and multi-phenomic diseases and blood diseases, okay? And the other thing is that it's comprised of different types of study designs, as I mentioned. Many of them are population-based studies that you've heard of, the Framingham Heart Study, which is one of the founding epidemiological heart studies, the Jackson Heart Study, but there are also studies of sickle cell disease, of asthma, et cetera. And in toto, there are about 155,000 participants, 155,000 in whom we at least have whole genome sequencing data right now. Now, there's another critical, very forward-thinking point about TOPMed, and that is is that it's not just about individuals with European ancestry. And indeed, 31% of the individuals have African ancestry, 15% are Latino. There's a reasonably large percentage of individuals with Asian ancestry, et cetera. And, so why is this so important? Well, if you going back to these polygenic risk scores for coronary artery disease, the largest initial study was actually done out of the UK and the UK Biobank. And this shows the results, the predictive accuracy of that relative to Europeans. So this was tuned, if you will, on Europeans, and you can see it works less well in Americans. And if you go all the way to the right side of the slide, it works horrifically in African Americans. And in fact, as Dan Katz reminded me during a call a couple days ago, many of these studies, in fact, actually excluded non-Europeans, right? And so I think you're going to see as we present some of our own data how well poised we are to explore these important questions, okay? All righty, this is a slide that maybe is of general interest. When I was training and maybe over the first 20 years of my career, I was kind of thought to myself, gosh, cancer research is really, really the place to be, right? There's so much being invested there. There's so many things that are changing in a positive way. It turns out that TOPMed is really catching up, and this is just one metric, the number of grants that are awarded. TCGA stands for the Cancer Genome Atlas, and you can see it started later and it took a while to catch up, but now it's growing at the same rate in terms of grants supported, and it's also dollars that have been invested. And also, so I just want to highlight that this TOPMed study is really bringing to the forefront cardiovascular disease and associated diseases. It can also be used for many other diseases that are in that umbrella, particularly in these large population-based studies, and it's supporting lots of grants. And I just want to highlight that it's an exciting time to be in the cardiovascular field. Now, there's a little bit of a discussion going on with these huge data sets. Should we be focusing on disease-specific analyses, or should we be focusing on kind of overarching biological questions and then kind of parsing disease in those individuals? And let me just give you an example. So there are about 200 proposals that are out there for about $100 million to look at specific diseases like asthma, sarcoidosis, heart disease, hypertension. And in this talk recently, Wayne Ugan suggested, maybe we should think about shared biological concepts, dive into issues, particularly in the COVID era, thinking about things like racial and ethnic disparities. And I'll give you one example. So, it's known for sure that African Americans have 20% more single nucleotide polymorphisms than Europeans, okay? And they have a very significant haplotype map. What does that mean and who cares? Well, we should care because, I recall back in about 2007, I was in a meeting in Dallas, and Helen Hobbs showed data from a Black woman who is part of the Dallas Heart Study, okay? And she is a 1 in a million person in terms of her human genetics, and it turns out she is a knockout, a human knockout for PCSK9, okay? And her LBL was something that I think Joe will be particularly jealous of, because I know Joe's a vegetarian. It was 11, Joe. So an LBL of 11, and that taught us a lot because she has a master's degree and she's an aerobics instructor. So it really says how low can you go with LDL cholesterol and why it's important to profile a heterogeneous group of individuals and do so in a comprehensive way, okay? And I think that that's important, and there are other biological concepts that people are interested in, of course, around age, around gender disparities, that these types of big data will allow you to address, I think, very robustly, okay? So getting back to this figure, where have we participated in TOPMed? I've circled the proteome and the metabolome. In terms of TOPMed, we've profiled I just give some examples: the Framingham Heart Study, the Jackson Heart Study, the multi-ethnic study of atherosclerosis, or MESA, We are deeply involved now with the cardiovascular health study, which is a study of aging. It's where all the frailty indexes were originally worked on. So that's or many of the frailty indexes. So we're very interested in that. So you can imagine of these kind of broad questions of gender, age, heterogeneity of ethnicity, we're really deeply involved in all of these studies. And by next year, we will have completed about 25,000-ish metabolomes, and we have completed just several thousand proteomes, but we are negotiating a large contract right now, and I hope to update you on that in the future. Okay, I'm going to say a few words about another really deeply phenotyped resource in which we're participating. And this is the MoTrPac Consortium. And I've shown some of these slides, but not quite in this context before. And this is called the Molecular Transducers of Physical Activity Consortium. And what it is it's an exercise training study. But it's a lot more than that. So it's 6 adult centers that are randomizing individuals to either an endurance exercise training arm or a resistance training exercise arm. And so it's about 3,000 people total, and it's normal individuals. Okay, that's in the 3rd bullet. And then as comparator groups, there's actually going to be some elite athletes that are being enrolled, about 150 who do endurance training, and then about another 150 who do resistance training. So it's going to be close to, that's 2,700 adults. There's also a pediatric arm as well, and that's something that's super important. You know, think of COVID research and why we need to study the entire aging spectrum, okay, and around exercises as well, of course. I'll just give you a few snippets of what the intervention looks like. So what you do is you get this very intense, you get really a cardiopulmonary exercise test, then you get this training test, and then you get a second cardiopulmonary exercise test a couple months later. So it's a single dose endurance exercise intervention. It's a supervised exercise intervention. You have to be within 5% of your targeted intensity. You have to adhere to the protocol. You have to make 10 out of every 12 sessions every 3 weeks. So it's 4 per week, as you'll see at the bottom. And the intensity starts off at 55 to 65% of maximal heart rate. And then it increases and it increases. And so it's about 30 to 40 minute bouts by week 3, okay? So that's the exercise intervention. What I'm here to highlight, though, is how deeply phenotyped these individuals really are. So I told you that there's these 2 cardiopulmonary exercise tests that are being done. A subject arrives, they rest for 30 minutes, they get their first muscle biopsy, then they exercise to peak, then they have another muscle biopsy. That 4-hour, because of, shall we say, some feedback from the participants, the 4-hour muscle biopsy was stopped, as you can imagine. But now there continues to be a day following muscle biopsy as well. And there's a series of blood samples, and in fact, 11 blood samples before, after, and in total, there's over 50,000 samples that are being obtained from these 3,000 individuals. But you can imagine there are multiple copies of it. There are actually over 1 million samples that will be sitting in a freezer at the University of Vermont. There's going to be genetics done, whole genome sequencing, RNA sequencing, proteomics, all types of proteomics, looking at post-translational modifications such as phosphoproteomics, metabolomics, DNA methylation. There's also there's tens of thousands of samples from rats that are getting exercise as well. Rats are the closest animal model to humans that can be tracked, they can be done in a tractable way. And then finally, a little bit cut off on this slide is the notion that what I really like about this is that we have these population-based studies, but we're going to be able to integrate and interventional, perturbational study as well. So you find something that's associated with cardiometabolic badness, if you will, in humans. And now if you want to then look to see, does that change with exercise? I think integrating that sort of information will be fantastic. And we have a number of programs that are related to this in our hospital. So here's the inflection point in the talk, and that is kind of, so what? What are you going to do with big data? Is it going to inform basic biology? Is it going to help us predict who will develop disease? Can we identify the mediators of diseases? Can we develop drug targets? Can we identify who will respond to a therapy? I mean, there are a million questions here, and I want to show you some really very new, unpublished, most of it unpublished data that really shows when you have these data sets, you can use them as immediate resources in many cases to get answers almost within seconds, okay? And I'm going to show you a couple of those, okay? And what we don't want on the bottom is one of these fuzz balls. I don't want to know some network. I mean, sure, at some level, we want to do some analyses of the connectivity of these things, but I'm kind of against that. I mean, as practicing physicians, we want to get at what is going to be clinically meaningful to us or biologically meaningful to us as scientists. And I think that that's what we as translational researchers can most contribute. So where are we going to start, okay? And the work I'm going to be showing today really is from Mark Benson and Debbie No and Zuzu Chen, who's an endocrinologist who works in the group, Dan Katz, Usman Tahir, and Dan Cruz, who unfortunately was cut off the bottom of this slide inadvertently. So where are we going to start? So there are a couple, if you start at 30,000 feet, you could think maybe of a couple simple ways to start, okay? And again, we're measuring a bunch of metabolites and proteins. We're going to focus on that today, that aspect of it in the blood. Again, think of blood LDL, but think of blood LDL on spades. Now you got 10,000 blood tests that you're looking at, okay? Wouldn't it be interesting to understand the genetics that determine those blood LDL levels or those circulating factors? So take out blood LDL and think about some other thing that we measure, N-type, BNP, okay, internal BNP. That's been measured by others for sure. But other things, blood coagulation factors, things that are important in biology, circulating cytokines that haven't been measured in thousands and thousands of individuals, and try to understand what the genetic underpinnings are, and could that tell us about drug targets? I'm going to give you one example of that. And then flip the coin, and this is a really fun part. People are beginning to associate hotspots in the human genome with lots of diseases, and we have no earthly idea how that works. But if you zoom in that gene and you know that there are a bunch of proteins or metabolites in some pathway that makes a lot of biological sense, well, that can tell you probably how that gene is working, right? And I'm going to show you some really interesting examples, or what we think are interesting examples that are relevant to present day malady. So I'm going to start with one kind of, positive control, okay? So it turns out by mass spec, we measure thyroxine particularly well, or T4. You measure that in a lab, in a hospital lab, by a radioimmunoassay or other measurements. And you can see this is one of these genome-wide association studies where across the x-axis, we've displayed all the human chromosomes, and on the y-axis is the minus log of the p-value, and I've circled in red for you that there's a little bit of a hot spot there that reaches statistical significance. And I was really excited because, hey, this could be an important new drug target for thyroid disease. And hey, that's an interesting protein, SERPIN-A7. I was super excited about this until I Googled it, and it turns out that SERPIN-A7 has another name, and it's called thyroid binding globulin. So you might say that's a bad thing, but it's actually a good thing because it means that our measurements are robust, that there's positive controls built in. It's what I like to call kind of anchoring information. And this first data that this was published as part of a cell metabolism paper a few years ago. Not that particular finding, but a bunch of novel findings. So I'm going to start now with one protein. You know, you see these Manhattan plots. We're starting to see what I like to call, and some of you have heard me say this before, these are more like Dubai plots, okay? And I'm going to focus on one finding of one protein over here, a super hot spot that's associated with apolipoprotein E, a circulating protein that we know is important in atherosclerosis. And in fact, you know, it's super important in human atherosclerosis. And in fact, to make a mouse have atherosclerosis, the only way to do that is to knock out ApoE. And Jan Breslow, who's down at the Rockefeller, did that about 30 years ago now. We also have known for about 30 years that one isoform of ApoE is a driver for Alzheimer's disease. That was found by Rudy Tanzi about 25 or 30 years ago. So now we have a circulating, super important protein that hasn't been measured in thousands and thousands of individuals. What the heck is controlling it? Well, one of the things that's controlling it, and that hotspot in the human genome, is, as we looked closer, is in an area of a gene, or right at a gene, called PPM1G, which is a protein phosphatase M1G. The reason I'm telling you that is because phosphatases and enzymes and G protein coupled receptors are the things that we target with our drugs, right? And in this paper, Mark Benson went on to show that on the left, I've just circled or squared the important boxes, that we were able to knock this gene out, which is highly expressed in the liver. And we were able to show on the right-hand side that we were able to decrease by about 20, 25% the amount of ApoE that's expressed by the liver. So now think about that over a long time. Could that influence atherosclerosis? So this is an important circulating factor in atherosclerosis, in Alzheimer's, and now we have a potential druggable target. Okay, that's published work. So now let's flip the story. Let's start with an important finding that of a genetic area of a genetic loci, or in this case, two genetic loci in a disease that's in the forefront of our minds. And can our data set inform what the heck might be going on there? So on June the 2nd, a paper came out in medRxiv, and we can discuss at length early publications, but this was what we thought was a pretty ironclad study that suggested that there were two loci that were identified for COVID-19 disease severity. And as I'll tell this tale, these went on to be published in the New England Journal. These are two loci, June the 2nd. On June the 3rd, there was an article in the New York Times that said genes may leave some people more vulnerable to severity of COVID IDs. It's just the next day. And this is the author, Frankie, who said, no one knows why, and that is haunting me, quite honestly, okay? So you got one locus, it's around the ABO locus, and another locus on a different chromosome, on chromosome 3. What the heck is it doing? And at 6:18 in the morning, I fired off a text. Did you guys see these report of these two loci? Can someone do a reverse lookup, which is exactly what I'm describing here? Start with the exact segment of the human genome and tell me what's associated with that segment of the human genome, because there are very few people, there are probably only 2 or 3 people in the whole world, I think, who have this data that can do it. So this is a software piece that Dan has adapted. This is Dan Cass's work, really nice, where we can kind of zoom in on what proteins or metabolites or any other factor that we measured might be associated with. In this case, we're focusing on the identical SNP that was identified in the New England Journal paper. And on the bottom piece is the zoom in on the local genes and some details about the local genes, and that's available information. But on the top, we've now integrated our own data set, and those dots are associated proteins, okay? And you can see at the top there I've circled the top association for that very identical locus that they published on June the 2nd. And this is now, I think, June the 4th or June the 3rd this came out. June the 4th, Dan is already looking at this, okay? And you can see that there's a p-value of 10 to the minus 300 that should get everyone, got our attention for sure. And that is a protein that's known as CD209, okay? So I'm going to tell you about it right now. So, you know, what's really gratifying is when you find something with that kind of p-value, and at least when you go to the literature, and it could make a huge amount of success, okay? So it turns out that CD209, as shown in this Cell paper, seems to be a novel determinant or an emerging determinant for how viruses attach to cells. Particularly, it's also known as DC sign or dendritic cell sign, we'll call it. Okay, so that seems to get interesting because the dendritic cells are what present antigens to our whole immune system. And it turns out that it was first described for flaviviruses, but then Gary Nabel and others have shown that this seems to be a very important binding protein for coronaviruses as well, okay? We're really excited about this finding. Okay, that's the top hit. These are a bunch of other proteins that are associated. I showed you a bunch of dots when we zoomed in. I'm just going to summarize these proteins by saying three things. Number one is there are other proteins associated with the same locus, okay? Many of those proteins are associated with thrombosis. And the strengths of the gene-protein associations vary between whites and blacks. And we think that that's fascinating as well, okay? All righty, so there's one other locus, super interesting. It turns out that that locus is associated with a secreted peptide called the chemokine, known as CXCL16, and its receptor, CXCR6, that's on T cells, okay? That we think is as equally as fascinating as well. So now we've others have searched the human genome just to drive this home. They found these two hotspots, and we're telling them two proteins. The first one is at the identical SNP. This is a few SNPs over, single nucleotide polymorphisms over, but it's at the very same locus for sure, okay? That seems beyond the play of chance for sure, okay? So just to recap, you know, on June 2nd, there's a paper that's published on medRxiv. On June the 3rd, New York Times article comes out. A couple days later, you know, I emailed our team and said, let's look it up. 5 days later, Dan Katz had generated a paper that we subsequently submitted the day following or 2 days later to a journal, okay? Now, because of COVID, we immediately contacted the team in Europe and we said, you can have these data. We'd love for them to be integrated in the paper, but it turns out they told us that their paper was very far along with the New England Journal, but suggested we contact the editors, who we then contacted, and to gauge their interest, they said, send us this as a letter and send us this information. That was back in June the 11th. Their paper was subsequently published online just 5 or 6 days later, and then published, and then subsequently, you know, out in print as well. Unfortunately, it's taken a little while to hear back from the editors of this journal. As of last evening, crickets, actually. It turns out I should have updated this because at 11 last night, as I was reviewing the final review of fellowships, I did get a ping that said the editor will be contacting you soon. Hopefully, we'll find out what that means. But I'll make one editorial comment in this time of COVID. The gratification, you know, we had to put these data out. We felt that this was super important. We did a lot of checks in our data. It turns out we had this finding by 2 different proteomic systems. We're 100% sure of our data. But it's a competitive world out there that's, you know, but we thought it was important to send it out. I'll just highlight, you do get things that make you feel good. These are from the associate directors of the UPMC cancer program, their cancer center there. I see you guys figured it out. It says here, great work and congratulations. Where are you submitting this? And it turns out we've been in contact with this individual. And they've been interested around this receptor in cancer. That I shudder to think to tell you about that story. But in any event, I mean, this is the feedback we've gotten, and this will be published, and people are increasingly using this type of information. And of course, they can have our data to look at it. All of the data that I've shown you are available or being posted on dbGaP. So all of our data are publicly, by definition, publicly available. Now it takes a little while to get up on dbGaP, but we can submit, we can provide all of this information. I'm going to give you one other example of this type of information, and then kind of get to the conclusion. So we've had a number of recent talks in the division around amyloid cardiomyopathy and transthyretin, and the importance of this mutation, particularly in African Americans, the V22I mutation that's in 3 to 4% of African Americans. 10% go on to develop, 10% of individuals with class 3 to 4 heart failure have this. That's from a JACC paper. And it's clearly an underdiagnosed cause of heart failure in African Americans. And this is paper from a New England Journal paper, manuscript suggesting, how important this is in several important cohorts. Okay. And these are data from our work in our lab, where Usman Tahir, who's now running our genetics program, said, again, let's zoom in and try to think about what might be associated with this disease. And clearly, it's important not just from a marker standpoint, but also from a marker of efficacy standpoint as well. We've heard from Kazi's work and Peter and others who have presented around this, and several journal clubs and clinical research conferences around new drugs to develop that target mRNA, but also tafamidis that target, and others that target the tetramer and this folding, and can we develop new biomarkers? I'm going to just cut to the chase here, that we have 2 novel associations with this very same variant. One for which there is some antecedent information, and one that is completely and utterly novel. So the top association in the Jackson Heart study is with a protein called RBP4, okay? And this protein, this results in reduced levels of RBP4. And it turns out it's been shown by others that RBP4 is a binding protein for transthyretin, which is the protein that contributes to these fibrils, okay? But what we have now shown is that reduced levels of this protein, so people have looked at this, if you will, in cells. We're showing this in humans in the Jackson Heart study, that levels are now seen prior to the onset of disease. And we have a number of associated hits in other signaling pathways. And I promise you I wouldn't show you too many fuzzballs, but there are other proteins that are related to this pathway for which we have coherent hits. We're making small fuzzballs, if you will, of related connectivity dots, if you will, of proteins, one of which is in something called the Wnt signaling pathway. And then we have this other very strong association with this metabolite that we're trying to identify. And you can imagine how this could be a disease marker, how this could be a marker of efficacy as well. Again, data that, now, I'm going to say it literally seconds to do this analysis. It took years and years, if you will, to develop this library, but fruits of this are coming, they're coming to fruition. Okay, so I'll end with a couple slides. They're a little bit of hardcore genetics, but I think it's important, and some of you have seen some of this, but it's important to say, how are we going to triage? How are we going to focus? We got 10,000 blood measurements There are 3 billion base pairs in the human genome. We have hundreds and hundreds of clinical phenotypes. We already have thousands and thousands of highly significant associations. How are we, what's the general paradigm that we're going to use to kind of put, how are we and several other groups kind of putting all this information together? So let's turn back to blood LDL cholesterol. And I've shown this slide before, but again, in a different context, in trying to drive home what we're trying to do. So, you know, in 2002, a French group identified variants in a family that were associated with, in this case, lower levels of, in this case, higher levels of blood LDL, modified levels of PCSK9. That group, in turn, it completed what's called this triangulation, also known as a Mendelian randomization experiment, where you simply ask yourself on the left-hand side of the triangle that you're looking at, is that variant associated with an important phenotype? And then in turn, we know that phenotype is associated with myocardial infarction. Is the variant associated independently with that phenotype as well? Does that whole triangle hold true? Okay. And what we're doing now is just swapping out, you know, LDL cholesterol for some new analyte that we've measured in the blood and saying, can we complete those triangles? And that doesn't mean that the other associations couldn't be useful biomarkers, but these are the things that we're going to start with in humans and then go back to model systems to dive into, okay? And that's how we're doing it. And I think we can do, we're generating this data, we're analyzing this data, and then we're going back to model systems to test for causal relationships. And of course, we're collaborating with wonderful large groups that have lots of expertise in statistical genetics, in trans-ethnic meta-analyses across races, et cetera, et cetera. So I'm going to give you one example of a finding that we had now a decade ago. And this was from a Nature Medicine paper where we identified abnormalities in amino acid signaling that happen over a decade before you develop diabetes. And this is probably the most robust, I mean, metabolomics finding that's out there. It's been found in whites, interestingly in blacks, in Southeast Asians, in every population. Now, to varying degree, and that's an important area of future investigation, but really it's the most robust finding, okay? And if you have these high levels, you're associated with sort of, if you're in the highest quartile, 2 or 3-fold increased risk of developing diabetes, even if you adjust for everything, your baseline blood sugar, your triglyceride level, what you weighed. We even had results in many individuals of an oral glucose tolerance test. So it stood the time of biomarker analysis, okay? But what we really want is it Carter horse, right? Is it driving disease? Okay, and I remember presenting this to a group of illustrious scientists, including Joe Goldstein, at the Broad in about 2008, who said, yeah, it's just a marker of insulin resistance. It's probably just a marker of insulin resistance. Good job, you've got an early disease biomarker. I presented it to another conference. Another National Academy member was even more, said, this is definitely, you know, you got an epiphenomenon here. I mean, and didn't even recognize that this could be useful as a biomarker. Okay, so it turns out that another group has actually followed up on this. We have as well, but this is work from a woman named Claudia Langenberg, who's a marvelous statistical geneticist who leveraged the UK Biobank to dive into this. And she drew her triangle slightly differently, but let me just walk you through it kind of briefly. She looked again, and you can see in the bottom left of the screen, of genetic variants that were associated with what are called branch chain amino acids. And she focused on these. These are the best predictors: isoleucine, leucine, and valine. And then she said, well, whereas Robert and Tommy's group, were they right? Was that associated with type 2 diabetes? and then they did this arm of the triangle, okay? And so they started off with this part, and they said, are the branch chains associated with incident diabetes? And that was, you know, p-values of 10 to the minus 16. They corroborated our findings, okay? Then their data, and actually our data, found variants in a gene called PPM1K that was associated with branch chain amino acids. And the reason I'm telling you this gene, it turns out that it's the gene that activates what's known as branch chain ketoacid dehydrogenase, which sounds like an enzyme that's important in branch chain amino acid metabolism. So I just want to drive home that a lot of these findings make biological sense. They find an enzyme that's important in the breakdown of branch chain amino acids, and lo and behold, those same variants are associated with type 2 diabetes in 300,000 individuals, or more than 300,000 individuals, as seen on the kind of right-hand side of this angle in diagram, which is a huge meta-analysis in the UK Biobank, et cetera, suggesting that this is not just an epiphenomenon, but the genes that control amino acid levels are important actually in the development of diabetes. And I know that a number of drug companies, including one that we're collaborating with, and I have no conflict here, I have no stake at all in that, but they are looking at drugs that block this pathway in diabetes. It turns out this pathway is also very important in heart failure as well. Yibing Yang at UCLA has shown that, and others have worked on this pathway as well. So I'm not saying that it could be two arms. There could be a biomarker arm and a pathway arm that's contributing to disease, but this is how we're marching through these thousands of associations with data that are now already generated. So I'll end with some final thoughts. I think that these, large-scale multi-omic profiling efforts are rapidly coming to fruition. I think, they're being generated. Integration of these data sets is beginning to illuminate new disease biomarkers and pathways, some of which might even be targetable for interventions. And there are many groups that are now diving into this. Human genetics is helping to prioritize these findings, but again, I'll underscore that I think that in terms of disease markers, we need to put our clinical thinking hats on. Are there other simple things that we can measure, or are there other genomic things that we can measure? But ultimately, I think also that studies in model systems will be necessary to definitively address causality. And we're doing those studies with knockouts and transgenics in our own model, in our own laboratory. And then one thought for the fellows that are in the audience, for the training and for the trainees, there's an enormous need and opportunity to help put these findings into the appropriate clinical context, okay? You can be a statistical geneticist all you want, but until you say, wait a second, that thing you just measured is something that's important in COVID-19 or in pulmonary embolus or acute coronary syndromes. You need that kind of wherewithal, and to say, well, we're already measuring D-dimer, does it add information on top of that, et cetera, et cetera. There's an enormous opportunity from the very basic science for those who are mathematically inclined to those who are clinically inclined. And there's really enormous opportunities across the entire spectrum for all of you. So I'll end with one final thought. We here in the Northeast are trying to emulate, the good things that are around us. These academic trees, there are these trees of knowledge and coaching, if you will, that have been generated. This is the Bill Belichick coaching tree. It's an impressive tree. I won't give you my personal comments about Bill Belichick, but it is an impressive tree of individuals. The work that I showed today, our own lab tree, could not have been done with a number of individuals in our group. This slide was put together, the notion of this was worked out with Jeremy Robbins. That is the goofiest picture of Jeremy I've ever seen. but these are a number of the individuals with whom we're collaborating. I think I've mentioned just about everyone along the way, so who've contributed to the work that I discussed. So thank you very much. And I know we only have a couple seconds for questions, maybe one or two, but thank you, and I'm happy to answer them.
Thank you so much, Rob. So if anyone would like to come off mute and ask one or two questions. In the meantime, we did have a couple of comments in the chat. Dr. Julia Resti had noted, for your exercise endurance study, you could look at MR patients with increased cardiac output, decreased systemic resistance, 24 hours per day, then examine the blood after MR treatment with MitraClip or valve replacement. And Jeremy had noted that MoTrPac and Heritage are in otherwise healthy people, but of course it would be very interesting in looking at exercise-induced remodeling in those with disease.
So stay tuned, Julie. It's a fantastic question. Jeremy is going to dive into that in a future grand round. I didn't want to take too much of that, but that's of course a key point, that's in normal individuals. That study is in normal individuals. Connie and I and Reza wrote a grant. They got a good score, and we're going to have to put it back in. And with Jeremy around, how about people with just cardiometabolic disease, MR? Think of all the perturbations. And we were discussing, Jeremy and I were discussing that the hospital is the place to do this, right? And I think valvular heart disease, we've looked with some of my former colleagues at the MGH in aortic stenosis before and after TAVR, for example. Great idea. All those kinds of, it's all on the table, and it needs to be done. And those are going to be ancillary studies that we're all working towards. And we were just talking yesterday, how about peripheral vascular disease? The wonderful program that Brett and Eric and now Alec are developing here. It's such a black box, and we can all think of many others, of course, as well.
Wonderful. Well, we're right at 9 o'clock, and given that we have cardiology fellowship interviews this morning, we will wrap things up. Thank you very much to everyone, and Maren has just sent out a link to note attendance for CME purposes. So, thanks, everyone. We'll see you next week.
Thank you.